Okay, so last time, right, we looked mainly at the production of a B cell receptor, which is another name for it is actually an antibody, and how they rearrange their DNA to form that receptor. So for the B cells, this happens in the bone marrow. For the T cells, as we just saw, happens in the thymus. So I have a link to, on our pages, web pages and animations and stuff for Unit 3. I have the link right here, Tcell Development, which goes to this animation right here, which I love this animation except for one thing. It doesn't label the MHCs that it was talking about, right? So I'm going to fast forward to that point where they start talking about that. So here's our T cells, and right here, they do label at one point, are the T cell receptors. And then they have two markers on them, CD4 and CD8. And this allows them to communicate with the two different classes of MHC that our antigen-presenting cells use to present antigen. MHC stands for Major Histocompatibility Complex. And so these are the molecules actually that create the problems when we try to do organ and tissue transplants. The immune system can't communicate with them, and so therefore thinks they're foreign and attacks them. So this very important education that happens for our T cells that communicate with our cells happens in the thymus as they're maturing. So let me get to one where they have the interaction. There we go. Getting closer. Okay. So, let me play for a second. Let me pause it. I was too slow. Still missed it. That's the spot. Okay. So when the T cells are first born, you could say, they have both of these receptors, a CD4 and CD8. And then their T cell receptor, which is what is interacting with the antigen, another thing that's not labeled here, right? So this would be the antigen. This molecule right here is the MHC. And so this is MHC class 1. And so it usually only interacts with CD8. And so that's why, since this one is recognizing, it's going to stop producing CD4 and it's going to go down that line of being what we call a CD8 T cell. And the importance here is that they're communicating with MHC class 1. Okay, so all your cells, all your nucleated cells, can present antigen on MHC class 1. So this cell is going to be able to communicate with just about any cell in your body. Unlike the other interaction, which is CD4 to MHC class 2, get a picture of that one. All right, so there we get to this one. So in this case, pause, Erica. There we go. Okay, so in this case, it's a CD4 it's recognizing, and that's because this is MHC class 2. This is a special class of MHC. This is only found on specialized white blood cells that can do what we call antigen presentation. These are your dendritic cells, your macrophages, and your B cells. And so as you see later in this animation, right, they interact with those cells. Dendritic cells and these guys also present MHC class 1, right? So all your cells do MHC class 1, but these guys are specialists in that they do class 2 as well, right? Presentation. So down here, right, if they're recognizing at this point dendritic cells, macrophages, right, they're presenting our own antigens, right? Our own proteins and stuff like that. Any of those T cells that recognize us, we do not want to survive, right? They are eliminated. Only ones that don't recognize us will be allowed to continue on. So that way, anything foreign gets into our body, they're going to recognize it and they're going to respond accordingly. Still with me? So the same similar thing happens for B cells, except it's not quite as complex as it is for T cells, because there's just only one type of B cell. And the important thing for B cells, again, like here, you don't want B cells, you don't want their antibodies, their B cell receptor recognizing our self-antigens. Right? They'll eliminate them. So only B cells that don't recognize us, that will potentially in the future recognize foreign antigens, will be allowed to survive and leave the bone marrow. Where's my little button? There we go. So B cells, T cells, remember these are lymphocyte cells. And they acquire the ability to recognize distinct epitopes. So remember when we talk about an antigen, we're talking about even just little pieces on that protein or on that bacteria. Right. Once they've done and made their receptors, they're checked out in that process that we saw. For B cells, it's in the bone marrow. For T cells, a little bit more involved and happens in the thymus. So now we're going to switch back to antibodies because they're quite complex. There isn't just one type of antibody. There's actually 5 different classes of antibodies that we're going to discuss. Another name for an antibody you'll sometimes hear is immunoglobulins. And a lot of times it's abbreviated IG. Because they are globular, believe it or not, they're proteins for the most part. Immuno, of course, they're products of our immune system. So we're going to look at, and I'd be able to identify the different parts of an antibody. And you need to be able to label them or identify what part is labeled. We need to know the functions of the different major portions of the antibodies. So there's a constant region, and constant means it stays consistent, right? It's always made the same way. Unlike the antigen binding sites, that's where the antibodies are going to vary, where they're different. When an antibody binds to an epitope on an antigen, your immune system can respond in a number of different ways. And we're going to look at 6 different protective outcomes that happen when antibodies bind to antigen. So let's look at a general shape of an antibody. So this is, of course, the stick drawing of an antibody. Kind of like how we draw stick people, right? This is a stick representation of an antibody. So you have 4 different pieces of protein that come together. But in truth, 2 of them are identical. You have what we saw last time being formed, the light chains, and these are the shorter chains. So therefore they were lighter when scientists when they studied them and compared them. You have 1 on either side, and these are identical. These proteins are made the same. They're made from the same stretch of DNA. Your heavy chains are in white here, and they're much longer, hence why they're heavier. But again, both chains, identical. The proteins are exactly identical. So you kind of have 2 sides to this molecule, right? But both are mirror images of each other. So when scientists were studying antibodies, they noticed certain things happened when they interacted with antigens and with the body's immune cells. At the top here, sometimes referred to the arms of the antibody, where we have the A and B, is where they would bind to antigen, right? So these A's and B's could represent the epitope even to an antigen. The bottom portion here, denoted by C, was where products of the immune system, whether it was immune cells or complement like we talked about before, this is where they would interact with the portion of the antibody.
What would interact with?
Your immune cells or portions of complement. Remember we talked about complement system last time, right? And how the proteins would bind. This is the part of the antibody where they would bind in this region here. So as I said, blue represents our two identical light chains, and then we have our two identical heavy chains in green. When scientists were studying them too, they found different ways to break up this molecule. And one of the ways is they broke it at this joining section here, and it broke into three pieces. This A piece, this B piece, and this C piece. A and B, notice, are similar to each other. They're mirror images of each other. And as we said earlier, these actually bind to antigen. Where the C portion did not bind to antigen and actually tend to crystallize in their procedures. So the A and B fragments were known as the antigen binding fraction, which A stands for antigen, B for binding. Works out with our diagram too, right? Because it's what I have labeled A and B. So the F means fraction, right? A piece of. So the FAB regions are shown here. They're the A and the B. And notice they comprise of all of the light chains and only a portion of the heavy chain. Of course, the bottom, right, I said it crystallized. That's actually why it got the C label. This is referred to as the FC region. And you'll hear this a lot because, again, this is the portion that the immune system interacts with, right? So when we say a cell has an FC receptor, we're saying that it's actually able to bind to this portion, this region of an antibody. So you have two FAB regions and only one FC region. Still with me? All right. When the antibodies were being made, remember we said that we shuffle up the DNA. And this holds true for T-cell receptors as well. So that there's always a portion of the antibody that's different from one B cell to another, right? They each pick their lottery numbers, they pick their antibody, and they're pretty much stuck with that combination. That portion that gets shuffled of each one of these proteins is called the variable region. And it's shown here in green. So for the light chains, their first segment is the variable region. Same is true for the heavy chains. They have their first segment as a variable region. So remember, this is where, too, the antibodies are going to bind to antigen. So this is what's going to be different for each antibody. All right, so we call this the variable region. The constant region, on the other hand, is what stays consistent from antibody to antibody. So if you were to look at an antibody, right, its protein structure here in the blue regions would be pretty consistent. And the reasoning for this is if it wasn't, notice that this is the region where the light and heavy chains bind to each other. If this wasn't consistent, you wouldn't have these two chains coming together all the time, like you do. We got to have this bonding that happens between these two chains. So combined between the light and heavy chains, we have this variable region. So it's that combination, right, of light chain and heavy chain that gives us even more variability. This portion of the heavy chain, which is in the FC region, right, again, it's going to be pretty consistent. But this is the portion that's going to determine the 5 different classes of antibodies, the 5 different types of antibodies. So depending on what type of antibody this is, will determine what constant region. And for each group of antibodies, again, this is consistent. It's constant. That protein structure is going to be made the same way. So again, the immune system can interact with this protein and know what it's dealing with. So variable region and constant region. So if we were to label every little piece in this diagram with a letter, just to give you an example of how it may be presented on a test, and I asked you to indicate which regions were variable regions, right? You'd have to say A, B, C, and D, right? The first portion of a light chain, the first portion of this heavy chain, the first portion of this heavy chain, and the first portion of this light chain. So when I have it written here, I mean A through D, right? A, B, C, and D. All the rest, right? E, F, G, H, I, J, K, all the way through to L. That's all constant region, right? And then remember we have the 2 fractions. So this would be one fraction here, this would be the other fraction here for the FABs, the top portion. Where the antigen binding sites, right, that would be denoted by M and N. And that FC region, right, is down here, I through L. And again, it's this region, this area, O denotes, this is where your immune system would bind on. Right, where molecules and cells would grab onto this molecule. So here's a picture of, say, a bacteria, right? Remember this blue portion, as you can see here, like the arms, are the antigen binding sites. And so it's going to attach to the bacteria in this orientation. So the leg, right, or the stem sometimes referred to, the FC portion, is sticking out. You see that? So later on, cells like a mast cell, which have a receptor for the FC portion to a specific class of antibody we refer to as IgE, that's how it's going to bind to it. Right? It's going to grab this portion of the antibody, that stem portion, with that receptor. As you guys will learn later when we talk about allergy, IgE is the major mediator for people who experience allergies. And my name happens to start with the letter E. So I usually tell my students, you'll never forget IgE because Erica hates it because she has really bad allergies. I'm not fond of how many IgEs I have produced by my body against all the pollens out there that our grasses produce. It makes breathing through my nose not a good thing. So, again, your immune system is very complex, right? But it's organized, right? So it knows how to interact with these molecules. And these molecules interact with the foreign substances in a specific way, right? Right down to the molecular level. So as I said earlier, right, we referred to that fraction as FC actually because it crystallized. That's how it got that name. But those different regions have specific important jobs, right? The FAB regions are going to bind to antibodies. That FC portion is what's going to interact with the immune cells, and it's what's going to interact with immune components like complement. So now we get to the 5 classes. Most of the classes are that very general, simplistic Y shape that we've looked at, what we call a monomer. Some of them, though, can form multi-structures where there's multiple monomers together. And those two that can do that are IgM, when it's secreted from the B cell that turned into a plasma cell, and IgA, when it is secreted out of our body into our secretions. Anybody want to guess what we consider a secretion? What do you secrete from your body? Sweat? Mucus. Lots of mucus. Women? Breast milk. One more extra secretion. Tears, saliva, mucus, breast milk. These are all loaded in IgA. So this is a protective antibody, but it's leaving our body, right? Mucus we secrete, breast milk we secrete, tears leave our body, right? Saliva, believe it or not, remember the digestive tract considered external environment. And so in this case, notice that the FC portion is kind of bound up, right? These guys form dimers, where two of them hook together end to end by their FC regions. And then you have the antigen binding regions, 4 of them now, sticking out. So we don't really need the FC portion available, because remember, this is leaving our body. So our immune cells aren't going to leave and follow these guys. Complement system's not going to leave and follow these guys. So that portion of the antibody doesn't really get used for IgA that's secreted. But instead, we really want it to bind antigen. So having 2 of them hooked together, it's going to bind even more antigen, right? 4 antigen binding sites. IgM, not quite the same story, although you can see the FC portions all kind of pointing in. The immune system can still interact with this one. But again, Y5 monomers, right? Why a pentamer? What does that increase? Amount of antigens it can bind to, right? How many antigen binding sites do we end up having now? 10, right? That can bind to a lot of antigen. It's also a very big molecule, right? So the good news with this one is that it doesn't typically leave the bloodstream, and especially for women, this one does not cross the placenta, so it can't cause problems for babies developing embryos and fetuses. Now, on the surface of your B cell, that very first class of antibody that it makes is IgM. But this is actually embedded in the membrane of the B cell, the FC portion, and the antigen binding portion is sticking out. We've also found in recent history that IgD is also on the surface of the B cell, that class of antibody. IgG, when it's secreted from the B cells, it's in that monomer form. So is IgA if it's in our blood. But once it moves from the blood, right, and goes into the secretions, it forms this dimer. IgD, if it's secreted, it's this monomer. And as I said, we've found recently, too, it's on the surface of the B cell. IgE is secreted as this monomer. And then it binds to those mast cells, as I showed you, right, where they're holding on to the FC region of this antibody, and the antigen binding sites are sticking out from that mast cell. So IgM, it's the first antibody you make in response to an infection. If those B cells have been activated and proliferated and differentiated into plasma cells, and we're actually secreting this antibody. It's also the only antibody that the fetus can make. So even when you're a fetus, you're making antibodies. It makes up about 5 to 13% of the antibodies in circulation, right? But it stays in your blood, right? It's too big to get out. Its structure is that pentamers we see, right? 5 monomers hooked together, lots of antigen binding ability. But on the surface of the B cell, it is in its monomer form. B cells can sometimes be stimulated by antigen alone to produce this antibody, this class IgM. But again, remember, this guy has limited capability. He's going to stay in your blood. It's not going to travel through tissue fluids and things like that. So a lot of times, it's not needed to have T-cell interaction. So that's where it says T-independent antigens. So when we don't have T-cell help, right, we can produce the IgMs. IgG is the most prominent in your circulation in your blood. At any one time, it makes up about 80 to 85% of the circulating antibodies. It's found in the monomer structure only. The good and bad news with this one is it can cross the placenta. The good news is it can cross the placenta and help protect the baby. The bad news is, anyone know what condition it can cause when it attacks baby's red blood cells? Hemolytic disease of the newborn, right? And this is where the Rh antigen is present on the baby's red blood cells, and the mom doesn't have it, but has made antibodies against it. And her antibodies from the mom can cross the placenta and attach to the baby's red blood cells and cause the baby's own immune system to attack itself and destroy its red blood cells. This antibody is also present in the colostrum. Anyone know what colostrum is? It's the first milk that a mother produces. And it actually isn't very nutrient rich, but it's chock full of IgG antibodies. And so it's one of the main reasons why, as science has progressed and we've learned these things about breast milk, doctors have encouraged more and more women to go back to breast milk feeding instead of formula feeding, if they can, right? Because you give these antibodies to your baby, and especially in that first milk. It's all the things you've been immunized against, all the things that you've been exposed to in your lifetime up until that point. You make tons of copies of these antibodies and you give them to your baby. And your baby can absorb them during this very early period of development into their body. Their immune system takes it in through their gut. It doesn't digest it, doesn't destroy them. The only problem is, since the baby didn't make these antibodies, over time they will deteriorate and be eliminated from the system. But it gives the baby some protection in those early months from you before its system starts working and before they can become vaccinated. It is the antibody of memory. It is the one that, as I said, sticks around for a long period of time. So it can really help you out. Your body will continue to produce certain antibodies too. ADCC is an acronym. Stands for antibody dependent cellular cytotoxicity. And so this class of antibody is the only one that can be involved in this action of your immune system, where antibodies attached to cells can actually cause the destruction of those cells by another cell. A lymphocyte at that, one who's very good at killing, whose name is awesome, natural killer cells, right? So natural killer cells can bind to IgGs attached to cells, that class of antibody, and destroy that cell. But that natural killer cell is only destroying that cell because the antibody is actually recognizing that that cell is bad. And so that's a very specific interaction. IgA is actually the most abundant antibody you produce, but it doesn't stay in your body, right? You're continually secreting it in your mucus, your tears, your saliva. And when you're secreting it, when it goes into your secretions, it's in this dimer form. And where this really helps us out is it binds to antigens. And for an infection, whether it's a virus or a bacteria or even a protozoa or a fungus, like, they need to be able to attach to us, right, in order to cause infection. If they're stuck in our mucus and antibodies coat them and stop them from sticking to us, they're just going to get washed away with that mucus, washed away with those tears, washed away with the saliva. So the main job of this antibody is to stop them from attaching to us. Keep them in the mucus. Keep them out of our bodies, in our secretions. IgD is very little in circulation. It's very rare. It's found in the monomer form, and it's really only seen in mature antibody response, so when the response is long-lived. But as I said, new recent studies indicate, too, that it is actually present on the surface of the B cell as a B cell receptor, along with class IgM. And then you have IgE, and this one, too, very little in your circulation because it doesn't really play a role in your blood. It plays a role in other areas of your body. And so it can bind to basophils and mast cells. Right? Both of these have similar granules. The difference is basophils are in your blood. Mast cells congregate in your tissues. And as I said, involved in allergic reaction because the fact that they bind to these mast cells and basophils, they're already there. So once the first time you're exposed and your body becomes what we call sensitized and it produces this class of antibodies, these IgGs, these IgGs coat your mast cells and basophils in those areas where the IgGs were produced. So the minute an allergen comes in and it interacts with the antigen binding sites on this antibody, this mast cell degranulates. So it releases things like histamine, which causes what in that local area? Yeah, inflammation, right? So that's why, like, especially people like myself, right, that are highly sensitive, I produce a lot of IgEs against ragweed. Literally, within seconds of inhaling that, I've got inflammation in my nose, so much so that I can't even breathe through my nose. Within seconds. Because the antibodies are already on the mast cell. You're not waiting for the mast cell to come along and find the antibody. They're already coded in it from the prior exposures I've had to it. And if I hadn't taken any antihistamines ahead of time, right, I can't stop that reaction from happening. That's why, for me, I take it every single day. Because I can't avoid, right, these allergens. If you're allergic to something like a cat, right, you probably don't own one, right? But if you have a friend who has a cat, right, take your allergy medicine before you go to their house, right? So that's already in your system, so that way it can help combat it when this reaction happens, because it's going to happen. Right? You already have those IgEs. And the bad news with that, too, especially with food allergies, the more and more you're exposed, the worse the reaction can get, the more severe the reaction can get. And the bad news with food allergies is that this is happening in your blood, right? Because you eat something, where do your nutrients go? Into your blood to feed all your cells. Well, if you have an allergen floating around in your blood, then you're going to have inflammation. You're going to have this reaction happening in your blood. Right? And this is why they can go into anaphylactic shock, because they're not getting blood flow to the vital organs, because it's getting all coagulated up and messed up and inflamed and everything, because the body's responding to something it thinks is dangerous, when in fact it is not. Right? Peanuts shouldn't kill you, but for some people they will. And it's because your immune system is thinking it's bad. So although this system is pretty awesome, right? That's definitely one example of where it could kind of go awry. So why do we even make IgEs? Right? Seems like they're just bad. Right? But, you know, they do play and could potentially play roles with other types of pathogens, especially like parasitic worms and stuff. So there's some reason for IgE. The unfortunate thing for some people, it can be a bad thing. So we have our 5 different classes, right? And for the most part, we're looking at monomers, right? But we have some that can form these much larger structures. That's our IgM. What's wrong with me today? I don't know. And one of the ways I help students and how I've remembered this one in the past is I always remember M for mega big, right? This guy is IgG is your main one, but your IgM is your mega big antibody, right? I don't have any tricks for IgA though. If anybody has any, share it with us. Right? That's your dimer, that's your secretion one. So when we look at, well, okay, so we produced antibodies. And we've looked at one instance with one type of antibody, the good old IgEs. But why do we produce all these other antibodies, and what can they do? Well, the main players are IgA, again, in our secretions, IgG all throughout our body, and IgM in our blood. Each one of these can do what we refer to as neutralization. And that's where when the antibody binds, it stops the pathogen from being able to do what it wants to do. And in the case of viruses, if you're coated in an antibody, you can't attach to receptors on our cell surface, right? You can't do the very first thing you need to do in order to cause infection, and that is attachment. So we've just essentially neutralized you, right? Because a virus does nothing outside of a cell, right? Nothing. It has to gain entry into the cell and use that cell's machinery to make more copies of itself. Toxins, too. They have to attach to our cells, right, in order to cause injury to the cells, or even sometimes, most of them, too, actually gain entry into your cell. But the first thing they got to do is attach. If it's coated in antibodies, can it attach to you? No. We've blocked it. We've neutralized it, right? We've stopped it from being harmful by stopping it from being able to attach by having the antibody attached to it first. And this is crucial to our protection against tetanus, because it's actually the toxin that you get vaccinated against, and you make antibodies against the toxin. And this is what your antibodies do, right? They surround it and stop it from getting into your cells. So each one of these classes of antibodies can do that. IgA, IgG, and IgM. IgA in your mucus, IgA in your mucus, right, in secretions, IgG all throughout your body, and IgM in your blood. The next one, right, again, preventing adherence. And this is really important, again, in our mucus. So IgA is this is one of the main reasons we produce it, is to attach to them so they don't attach to us. That's not necessarily going to neutralize a bacteria, though, right? Okay? We're just stopping adherence. That's not necessarily going to keep them from getting to us. The other thing we can do is when it attaches, it can prevent them from moving, especially if it's attaching to their flagella. Because just as if you're putting down the bayou, and this has really happened to me, right? We're going down the bayou doing research, one of my colleagues, all of a sudden we're not going anywhere. We pull up the motor, there's a sweatshirt wrapped around the propeller. Okay, that's why we're not going anywhere. So we cut the sweatshirt off, and off down the bayou we go. Well, imagine the same thing, right? Bacteria flagella move like a propeller on a boat. They swirl around. Well, if you attach antibodies to it, they're going to get it all gunked up, right? And it's going to stop being able to move. Except the bacteria aren't going to be able to go and cut off the antibodies, right? Like we did the sweatshirt. It's going to gunk them up, and they're not going to be able to move. We're going to immobilize them, right? So antibodies, you know, just that binding action can really be a pain in the butt for a pathogen and very helpful to us. And this can happen in our mucus with IgA, or it can happen in our blood with IgG. And think about that one, and IgG, and think about IgM when it binds. It's huge, right? A whole bunch of those binding to the flagella. That flagella would not be going anywhere anytime soon. The next we look at is the fact that, and especially with just a single monomer, right? We have two antigen binding sites, right? Those two arms of the antibody. So at the very least, an antibody can actually hold on to two pathogens at the same time. And then when you have more antibodies, you can get this cross-linking effect happening. Now, when this happens for large antigens, we refer to this as agglutination. So when bacteria, which are relatively large, right, get cross-linked, they get clumped up. This is referred to as agglutination. Now, a small thing that's soluble, like say a toxin, when the antibodies bind, it's going to make it a much larger complex of molecules. And if you had it in a liquid solution, it would precipitate out. And that's why it's referred to as precipitation. Because something that's soluble could be in solution, right? You wouldn't see it. And I kid you not, we do immunological tests outside the body where we use antibodies to detect the presence of antigens. We're talking about molecules. You can't see those with your eyes, right? But if you add enough of them, antigen and antibody, say in a test tube or in a gel matrix, when they come together, you can actually see that formation of that larger molecule. You'll see it precipitating. You'll see this white line appear, right, as all these molecules are binding together because that antibody is recognizing that specific antigen. How many of you guys have taken AMP2 lab? Anyone taken that yet? Okay. So you did agglutination, right? What did you agglutinate with antibodies? Red blood cells, right? Because red blood cells have different markers on their surface. Different people produce different types of antibodies. One of the ways we use to detect, that's the way blood typing is done all the time, is we have antibodies we know bind to specific antigens, right? So we know this antigen binds to the A blood type. So when you add it to blood, it binds to those red blood cells and it clumps them up. So much so that you can see it happening right on a microscope slide or in a little plastic well. Right? My blood is boring though, because I have O-type blood. I don't have A or B antigens. So when you add those antibodies, nothing happens. Right? So one of the other places that I worked, the nursing students were always, you know, doing practicums and stuff like that, and they'd have to volunteer, and they'd ask for people to come give their blood so the students could get practice running these tests. And I'm like, yeah, I'll go, but mine's not very exciting. But it still gives them the opportunity to run the test. Now, they typically wouldn't look for the RH. That one, antibody doesn't stay stable as well because, again, it's a different class. The one you make against ABO blood group is the IgM. It's nice and big. And that's why you really see that clumping really well for those particular antigens. But that's also why you don't have to worry about matching your ABO blood type to your baby. Because remember, that one's not going to cross the placenta and attack your baby if your baby's different from you. It's the RH that you have to worry about. And I'm RH positive, so mine would clump up really good. Yeah. And so the other thing is, remember the Nobel Prize website, the very first website for immunology that I sent you guys to? they have an awesome blood typing game, and they've even improved it over the years so that, you know, immunologists like myself are happy because, like, they look like antibodies now when you look on the test tube instead of these weird little characters they used to have. So really impressive and really can help you learning that material if you're struggling with it. It's a pretty cool site. So why would that be beneficial, like, say, inside of our body? Why would we want to, like, clump them up like that? Yeah, neutralize them faster and kind of keep them all in one area, right? And if there's a whole bunch of them together, it'd be much easier for, like, our macrophages to grab onto them and destroy them. And that's one of the next things that we're going to look at is cells actually interacting with antibodies, right? So far, right, there was no immune cellular interaction here, right? No immune cellular interaction so far here or here. Here, that special ability I said, remember, for only IgGs, that antibody-dependent cellular cytotoxicity. So if you have a cell that's displaying on its surface foreign antigen and the antibody recognizes it, the antibodies can coat that cell. And if it is IgG that's coating that cell, when a natural killer cell comes along, it has an Fc receptor for IgGs. It will bind to it, and it will send a signal and toxic substances to kill that cell. And so this is very beneficial too, like if our cell gets infected with a virus, right? And the virus proteins are getting put into the membrane to make their envelope, right? And then we have antibodies that come along and attach to those viral proteins. Natural killer cell comes along and will kill that cell. So that's good old, right? So that one's special. Antibody-dependent cellular cytotoxicity. Only IgGs can do that with natural killer cells. And again, on that same principle of the Fc receptor, when antibodies bind to something like a bacteria, phagocytic cells like your macrophages and dendritic cells have receptors for the Fc portion of your antibodies. And they're going to grab onto that and engulf it. And remember, when we tag something and a phagocytic cell comes along and engulf it, that tagging process is referred to as opsonization. Do you guys remember what complement protein also opsonizes, binds to the surface of bacteria? Phagocytic cells come along and grab it up. So C for complement, right? 3 is the number, and B is the fragment, and I always remember B for binding. C3b binds, opsonizes pathogens for phagocytosis by our white blood cells, like macrophages and dendritic cells. Guess what, y'all? It's right there on the slide in front of us. Opsonization by C3b. That's one of the outcomes when complement becomes activated, right? Does anybody remember what it's called when an antibody binds and complement proteins bind to it and activates the complement cascade? Is it alternative, classic, or lectin binding? It's a classical, right? It's what they discovered first. No, classical. The classical pathway of activation of complement. Complement can be activated 3 different ways. The classical pathway, which is where antibodies bind, and then complement binds to it. Alternative is when C3b binds. Lectin binding is when you have those lectin binding mannose molecules bind and they activate complement. And remember, when complement becomes activated, you're going to get the production of more C3b that can opsonize even more cells. You'll get the inflammatory response. You guys remember which 2 did I tell you we need to remember for inflammatory response? C3a and C5a. So they're going to make the blood vessels leaky, right? Those complement proteins are inflammatory cytokines. They're going to cause the dilation of blood vessels and that movement of the neutrophils to that area. And then cell lysis can happen. And how does that happen with complement proteins? What do they do? What do they form? Yeah, they form like a donut shape, right? But what is that complex called? MAC. But what is it? It's not a hamburger. Yeah, a hole in what structure? The membranes. Remember, membrane attack complex. Right? So it's a whole set of those complement proteins that form that donut shape that inserts into membranes. So remember, who did we say was more susceptible, gram-positive or gram-negative, to this type of attack? Negative, because what do they have? An outer membrane, right, that those can insert right into. So one more level of detail. There's only 2 classes of antibodies that can activate complement. Anyone want to gander a guess? What are the 2 most common antibodies you have in your body? IgG and Mr. Mega Big IgM. And as I said, when antibodies activate complement, that's referred to as the classical pathway. The alternative is C3b binding, but in all reality, it's probably what happens more often than classical. Because classical, you got to have antibodies. And as we'll find out, it takes a while to make an antibody. You're not going to have those right on scene, right when a pathogen gets in. So these are the 6 possible outcomes. We can get opsonization, so we get destruction by a phagocytic cell. We can get complement activation, which can lead to more opsonization with C3b, inflammatory response, more white blood cells to the area, or lysis of those foreign cells by the MAC complex. We can immobilize and prevent adherence with the use of antibodies. We can cross-link them up, and we can agglutinate or precipitate depending on what type of antigen we're talking about. We can cause natural killer cells to kill it if it's coated in IgGs. And we can neutralize things like viruses and toxins by having antibodies binding to them and stopping them from doing their action. So, and as I mentioned earlier, right, IgM is really good at agglutination, right, because of how many antigen binding sites it has. So here it's binding to. One of the nice things I like about this drawing is, again, notice that this IgM is binding to a specific epitope, right? Little piece of this bacteria. And then this antibody is binding to a different one. And this is actually showing you what? Red blood cells, right? And how the IgM really can clump up. It can grab onto, 5 at a time and really clump them up, really agglutinate them. Oh boy. So this is how antibodies should be attaching to a bacterial cell, right? Or an antigen. And so we have our FC portion sticking out so that our phagocytic cells can grab on, or maybe a natural killer cell can recognize it. Maybe complement can activate. But there are some bacteria out there that have developed receptors that grab onto the FC portion of our antibodies. So they grab onto the portion our immune system is supposed to interact with, flip our antibodies in the wrong orientation. So our immune system's not going to interact with this FAB fraction, with this antigen binding fraction, right? It should be interacting with the FC portion. It can't even see it because this guy's gone around and grabbed it. Staphylococcus prurigens that causes strep throat does this. It's one of its protective ways it protects itself. It literally picks up any one of our antibodies by its Fc portion and just coats itself in our own antibodies. So we can't get at them. So like I said before, right, even though antibodies are pretty awesome, right, and have amazing abilities, the reason why we still get sick is some of them have found ways around our protection system, right, or can even use our system against us in this case. So let's go ahead and take our break. And